everyone, and welcome. This is Tom Maslanik welcoming you to the presentation today. We are hosting this talk from Immunology 2015 in New Orleans and streaming live to those of you online with Bite Size Bio. As a reminder, we will have a brief Q&A session after the presentation. For those of you streaming the webinar live, please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I will address them at the conclusion of the talk. This talk is, of course, sponsored to you by Bio-Techne, the parent company of Novus Biologicals and R&D Systems. Bio-Techne was conceived by R&D Systems when we realized that there was an opportunity to become a better partner to our researchers by bringing together additional companies to provide more complete solutions for scientists. In addition to the proteins and ELISAs that we are known for, we now offer innovative instrument platforms through Protein Simple, which allow for simpler Western blotting and more sensitive ELISAs. We carry small molecules, which researchers have trusted for decades through TOCRIS. And by combining the R&D portfolio of antibodies with Novus Biologicals, we now offer more antibodies than ever before. So for you flow cytometrists out there, our catalog of conjugated primary antibodies covers over 5 times more targets than any other antibody supplier, so you can achieve more sophisticated characterization of better defined populations of cells. Our conjugated primaries are backed by R&D Systems' promise to provide highly specific reagents with industry-leading lot-to-lot consistency, as well as Novus's mission to facilitate better multicolor staining by providing 17 fluorophore options for each monoclonal we conjugate. So today, we are excited to highlight the work of a researcher we are privileged to work with, Dr. John Nolan. He will be presenting his talk titled Measuring Extracellular Vesicles by Flow Cytometry, Challenges and Prospects. As many of you know, his lab is developing flow cytometers that make leaps forward and overcome a number of limitations of the current technology. His development pipeline includes cytometers designed to facilitate greatly enhanced multicolor flow cytometry, as well as to remove barricades that hinder our ability to measure individual small particles like exosomes and microvesicles, which is something I'm very interested in and of course the focus of today's talk. I met John for the first time at the La Jolla Immunology Conference a few years ago when I was searching for information on a spectral flow cytometer his lab was developing, and his vision for the potential for greatly enhanced capabilities of flow cytometry really drew me in, and I continue to be excited by the implications of his work, as much of the scientific community. John's career has been punctuated by numerous publications, grants, and accolades, including a fellowship with the American Institute for Medical and Biological Engineering, and a presidency with the International Society for the Advancement of Cytometry, or ISAC. This is really a man that needs no introduction, but since I had the opportunity to do it today, I wasn't going to miss it. Everyone, please join me in welcoming Dr. John Nolan.
Thanks, Tom. Thanks for inviting me to come talk about this interesting new area in biology, extracellular vesicles, in particular about some of the challenges and opportunities for measuring them. To convey the idea that this is an area that's in need of technologies to enable the measurement of particles. And while the jury is out on exactly what the technology would be, we're starting to see some clarity based on work in the past few years. So here's an overview of what I want to talk to you about. I'm just going to give a brief discussion of extracellular vesicles and highlight the challenges to their analysis. I'll briefly review some of the existing approaches to measure these particles. Then I'll focus most of the talk challenges, particularly for flow cytometry, to do these. And at the end, I'll close up with some resources that you can use to hopefully improve your research. So it turns out that all cells, all living cells, release vesicles constitute as part of normal everyday life. Vesicles can come from several sources in the cell as a result of several different biological processes. Certainly when cells die or undergo apoptosis, vesicular debris are released from the cells. But those aren't the particles I'm going to talk about. I'm going to talk about exosomes and ectosomes also known as microvesicles. These are small vesicles that are either secreted from internal stores of the cell, these are exosomes, or are shed from the plasma membrane. It's ectosomes, microvesicles. So all cells can release these vesicles by different mechanisms. They have different compositions, it's clear. So they're complicated. Then if you look in a complex fluid like blood, situation's even more complicated because many different cell types are seeing these vesicles. So in blood, there are vesicles from every cell that has access to the vasculature, plus vesicles from different origins within those individual cells. These can have widely different physiological consequences, and so understanding them, is potentially a new window into physiology. Just a little closer look at the differences between ectosomes and exosomes. So exosomes are formed by inward budding of the endosomal-like compartments of multivesicular bodies. So we can have organelles that have within them vesicles. When those organelles fuse with the plasma membrane, those vesicles are released into extracellular space. Vesicles can also bud off or bleb off from the surface of the plasma membrane. And there's good evidence that the composition of these two types of vesicles are different, even though they have very similar size and physical properties. It's important to note this is probably a simplification. There are probably subtleties to these two major classes of vesicles that we fully appreciate yet. And I think more study, it wouldn't be surprising to find out that there's more than just these two classes of vesicles. So the vesicles have been implicated in almost any area of biology that you'd want to consider. Just highlight roles in immunity here, immunology concepts. But for a couple decades, there's been interest in especially dendritic cell-derived vesicles in terms of antigen presentation. And been numerous efforts to exploit exosomes and exosome-like particles for vaccine development. But beyond this, immune-specific role of exosomes, exosomes and extracellular vesicles play a key role in all sorts of cell-cell communications, not just restricted to immune cells. So extracellular vesicles are interesting for a number of reasons. First of all, because they're released from cells, they potentially tell us something about the state of those cells. Obvious case would be if you have a tumor somewhere and that tumor is releasing these vesicles, you might learn about the type of tumor by analyzing the vesicles that are released in liquid biopsy. To the extent that these vesicles actually play roles in physiology, they're interesting as potential therapeutic targets. Then vesicles themselves have a potential to be therapies. They can carry molecules that can provide targeting. They can carry cargoes that have an effect on cells. And so there's significant interest in exploiting vesicles for therapeutic purposes. So those are all the upsides why people are interested in vesicles. What are the challenges? First of all, as I just mentioned, they're very heterogeneous, coming from many different cells and many different sources in the cell. And so this is, the way I think of it, is it's going to be at least as complicated as immunophenotyping lymphocytes in blood. But it's going to be actually more difficult because these things are so small and dim. They don't scatter much light. don't have many antigens on them. And while flow cytometry has been a popular tool to analyze these, in reality, conventional flow cytometry performs pretty poorly, as I'll try to explain. So just to drive home what the implications of having such small particles are, especially if you're going to try to do immunophenotyping by some approach like flow cytometry. Just consider the relative abundance of receptors on cells and then on a small vesicle. So I've just taken a monocyte here as a model cell. You could do this for your favorite cell of interest. We know about the average number of surface receptors on monocytes, for instance, CD14 being a relatively abundant cell surface marker, CD11A being sort of a moderate abundant surface marker, and CD16 being actually fairly low abundance, although still readily detectable at conventional flow cytometry. You just make the assumption that the surface density is going to be the same on the vesicle as it was on the cell of origin. You can calculate how many receptors you might find on a vesicle. So in this graph, I have the size distribution of vesicles from blood. The median size is about 100 to 150 nanometers. It's pretty well accepted. There are larger vesicles that might go out to a half a micron. Some rare ones that be even larger. The vast majority are less than 200 nanometers in size. So if you take these three high, medium, and low abundant surface markers and calculate how many you might get on a 100 nanometer vesicle, the answer is not very many. And so this is important to keep in mind as I discuss some of the issues associated with calibrating your flow cytometer and the flow cytometry measurement. So it's worth surveying the basic elements of EV analysis. First, recognize that your isolation of your sample have a big effect on whatever you measure downstream. I'm not going to focus on this here, but how you collect your blood or other biofluid, what sort of processing you apply to that to purify or enrich your vesicle prep. Those all have impacts on what you measure downstream. In general, you take the attitude that less is more when it comes to handling samples. So we try to avoid centrifugation steps and other sort of sample handling if we can possibly. But once you have your prep of vesicles, you have some choices about how to make measurements. Bulk assays, familiar with, like an immunoassay, basically measures the total antigen in a tube. Similarly, dynamic light scattering can give you an idea of the size distribution of particles. Genomic and proteomic approaches are important, but all of these provide an average measure of your sample, which, as we've already discussed, is a heterogeneous population of vesicles. So it really calls out for single particle analysis. EM is the gold standard for looking at nanoparticles. If you couple it with immunogold, you can learn something about surface protein expression, phenotype of those vesicles, but not really practical for large-scale studies. There are two methods to measure and count and measure the size of vesicles that I'll talk about a little bit. Nanoparticle tracking analysis and resistive pulse spectroscopy. So in terms of getting concentrations, these are useful methods. They're single particle based methods. But they don't really tell you much about vesicles other than their size and concentration, which is why so many people want to use flow cytometry. It's a single particle measurement tool. You can measure multiple molecules on the surface of a particle, a cell. And so, there's lots of good reasons to use flow cytometry. Talked about microscopy a little bit. The group of Alain Bresson in Bordeaux has done some really workmanlike studies on quantitative analysis of extracellular vesicles, blood, and plasma. So if you want to see a good example of a quantitative EM study, I would suggest you look at the work from his lab. But again, while providing useful fundamental information, isn't really practical as a routine research tool. Nanoparticle tracking analysis counts and measures the size of vesicles by measuring their Brownian motion. So by measuring how fast the particles move, you can estimate their size, their diameter, assuming that they're spheres. This is a very useful approach that, as a starting, as I'll describe, it's really important to know your particle concentration doing flow cytometry, and this is a good way to do it. Another good way to do it is resistive pulse spectroscopy. Think of this as a nanoscale Coulter counter. There's a nanopore with a voltage across it, and individual particles are counted as blockages in the current flow, the nanopore, and the size of the current block will tell you something about the size of the particle. So if you calibrate the instrument correctly, you can get size and concentration. So both of these are very useful tools, and we use them in our lab in conjunction with our flow cytometry methods. And I think it's probably a good idea if you're going to work in this area to have one of these methods at your disposal. Which sort of gets us to this question of how big are extracellular vesicles. Rink Newland's group in Amsterdam has undertaken a really systematic study of different methods to analyze vesicles. Here's an example of 5 different approaches, including NTA and RPS. And the consensus is that vesicles are between 100 and 150 nanometers, maybe going down to 70 or 75 nanometers, the tail going up to a few hundred nanometers. This is the major size distribution. There are exceptions. Some interesting accounts of cancer cells extruding very large membrane vesicles in a clearly active, regulated way. These have been termed oncosomes. Red cells, can be red cell fragments because red cells are so abundant in plasma. You're just going to naturally find a lot of red cell fragments. If you look at some of those EM studies, you'll find long, elongated tubules. But in general, these are fairly rare types of vesicles. The vast majority appear to be vesicles just like we draw cartoons of, sort of round balls. So right back to make the point of how many molecules are we looking to detect? And you can see that even for a highly abundant surface marker on monocyte, CD14, by the time you get to the main population of vesicles being between 100 and 200 nanometers, we're talking about less than 100 antigens. And that presents a pretty serious challenge to conventional flow cytometers to measure. So this is why we want to measure flow cytometry. Multi-parameter analysis is clearly going to be required. Flow cytometry has well-documented abilities to count particles and also to be quantitative in fluorescence that allows you to count the number of molecules per particle. So we're going to talk about some of the challenges of applying those things which people are familiar doing on whole cells to these small vesicles. It's probably just good to note quickly that people have been using flow cytometry to measure vesicles that have been captured on beads. This approach has been around for a couple of decades. You can buy beads designed for this purpose. You can buy kits designed to do these sorts of measurements. And basically, you can capture vesicles either non-specifically, just by a chemical reaction with the surface, or you can put a specific antibody surface and immunocapture that has some specificity, and then you'd have some fluorescent reporter cocktail to measure what you caught. So this is not challenging for conventional flow cytometry, but it's also not a single vesicle analysis. It's essentially a bulk measurement, whatever vesicles you captured surface of the. So I won't say any more about this. I'll talk about is the attempts to measure individual vesicles, and people have been doing this for a long time. The first accounts go back to the late 80s, and in any number of papers you can see plots that look like this, and words that go something like microparticles are defined as small 0.1 to 1 micron diameter vesicles released from cells, and that size estimate basically comes from the use of beads to standardize measurement. One of the first things people learn when they start doing flow cytometry is that light scatter is proportional to size. That's generally true, but there are lots of exceptions. And the bottom line is we can't use polystyrene beads to calibrate the size of small particles like vesicles. So in reality, light scatter doesn't measure size or granularity. Under certain circumstances, can be proportional to size, and that can be useful. In reality, light scatter is a complicated function of not only the wavelength of excitation, but the angle of light collection, particle size and shape, and also the refractive index of. So it's complicated, but it's been studied for more than 100 years, so it's actually fairly well described mathematically. And I'm going to show you some calculations I did using this program bplot, which you can get online. It's essentially a implementation of B scattering theory in a nice graphical interface, so you can plug in numbers and predict what scattering might be. And doing these, you can come up with some interesting insights. First of all, there's a difference. The angle of collection makes a difference, as does the wavelength. So above the wavelength of light, for our purposes, we'll calculate it at 488 nanometers. Above the wavelength of light, light scatter is highly angle dependent, and scatter is much stronger at the small or forward angles is at side 90 degrees. Of course, that's the angle there. You're looking into the laser beam, so typically you have an obscuration bar to block the laser, so you might be looking at, sort of 3 to 10 or 5 to 20 degrees the forward direction. They'll give you the brightest scatter. You notice this is a log scale here. Intensity of scatter falls off quickly as you change the angle. So that's why forward scatter for cells, for large things, is good. However, below the wavelength of light, for particles that are smaller than, say, 488 meters, angle of collection doesn't make so much difference. And because you have less background when you're looking at 90 degrees as opposed to looking in the forward direction, 90 degrees is generally recognized to be sort of more effective for measuring very small particles. Above the wavelength of light, you can get proportional increases in scatter intensity in response to size, or at least that if you have a bigger particle, it'll scatter more light. Below the wavelength of light, the behavior is different. There's a proportional dependence of particle size on light scatter intensity, but it's a radius to the 6th power. So every time you go down by a factor of 2 in radius, down by a factor of 64 in intensity. So things get dim really fast and things get small. So this is a real challenge for using light scatter to measure these particles. Final challenge to making light scatter measurements is the effective refractive index. So polystyrene has a fairly high refractive index compared to water. Lipid vesicles have a lower refractive index. It's much closer to the refractive index of water. Depending on who does the calculations, the rule of thumb we found is a vesicle will scatter 30 to 100-fold less light than polystyrene bead. So this is the reason we can't use beads to calibrate size of vesicles. Some people are working on trying to understand this and try to apply the math to how calculation of size from light scatter, but it's a challenging. So what can you do with beads? Beads are useful for setting reference windows, right? So if you think of beads as reference particles and not as calibration standards, you're probably okay in terms of being able to set up your instrument the same day to day, sort of be able to compare perhaps between labs. The ISTH, International Society for Thrombosis and Hemostasis, these are people who are concerned with coagulation, platelet microparticles in particular, the role in clotting, has spent a lot of time comparing light scatter measurements from different instruments, sort of interlab testing studies. They've published some work on this, and the basic conclusion is that it's hard to standardize across instruments and labs. Every instrument will measure light scatter a little bit different angles of collection, etc. So light scatter measurements are a challenge. But it's what most people learn how to do flow cytometry, so they trigger their detection off light scatter. And most of the papers use light scatter. This, again, another paper from Rink Newland's group, shows a comparison of how many vesicles are actually detected by different methods. In panel A, you can see the conclusion is that not very many vesicles are detected by conventional light scatter triggered flow cytometry. When you measure, if you do these measurements right, you're only detecting the very, very largest vesicles. So what are some of the pitfalls that this can lead to? So the big problem is that flow cytometers really aren't sensitive to do a good job. And this can lead to artifacts, irreproducibility, difficulty in standardizing. But I think a lot of these can be addressed by appropriately calibrating measurements. And so I'm going to spend a little time talking about how to standardize and calibrate your flow cytometer to aid in measuring these things. There's a bunch of other artifacts that have been identified by people looking carefully and doing careful quantitative work. One is the reasonably well-known occurrence of protein aggregates within your antibody fluorescent conjugates. I think even for lymphocyte immunophenotyping, centrifuging your antibody conjugates is considered practice. Not everyone does it. It's really essential here because those fluorescent antibody aggregates are about the same size and about the same brightness as an extracellular vesicle. So if you do the antibody-only control, you may find yourself be surprised at how many particles you might. So it's important to clear your antibody solutions before you use them. Filtration is obviously important for all your buffers. Anyone who spent time in the lab has been able to see calcium carbonate precipitate out of PBS when it's not handled right. But even when it is handled right, the addition of calcium to phosphate buffer can result in microprecipitates that will trigger light scatter and show up as an event, flow cytometry. probably want to stay away from phosphate buffers that require calcium in your experiment, for example, using an Xn5 as a and then one artifact that we shed some light on was the effect of coincidence on flow cytometer measurements. And I'll discuss that a little more here. So coincidence is well understood, from the earliest days of flow cytometry. It's when you have more than one particle in probe volume at a time. If you have a high concentration of cells, you're more likely to have coincident events. Happily, when there are coincident events, you can usually tell them apart because they have distinct light scatter, right? So you gate out the doublets and light scatter. It's not really possible with these vesicles because they're so small that it's hard to light scatter anyway. And for vesicles, often it's hard to know the concentration before you do staining. So if you're not sure of what the concentration is, may measure coincidence. I think this is pretty widely recognized now as a major and pervasive effect of the vesicle literature using flow cytometry, where individual particles may have been too dim to detect, but the presence of 10 or 20 or 100 vesicles in the probe volume will trigger detection of instrument and show up as an event that wasn't real. So coincidence is a problem. Marco Wabben's group in Utrecht has been doing some very nice systematic flow work using fluorescence-based triggering rather than light scatter-based triggering. And she's nicely shown what can happen when you have too high a concentration of particles. Draw your attention to the oscilloscope traces at the bottom. At one time, all flow cytometers had an oscilloscope attached to them. You could actually see the pulses of each event as it went through. Most today do not, but some do, and Marco's did. And so the blue trace on the bottom left shows a single pulse of single event in light scatter and fluorescence. But as you increase the particle concentration, you're still measuring particles, but they're not single particles anymore. They are coincident occurrences of many particles in the sample volume, which will leads to A, underestimation of your particle concentration, B, overestimation of your particle brightness, because you're really measuring many particles. And the experiment that can tell you whether you have this coincidence occurring is just to do a serial dilution of the sample. So if you dilute your sample by a factor of 2, measure it, and then dilute it by a factor of 2 again, and measure it, you should see the event rate go down proportional to the dilution. But brightness of the particle shouldn't change if you're really measuring single particles. And so coincidence is difficult to recognize, but it's so pervasive, you really have to look. And this is a good way to look for it, is to do serial dilutions for particles and see if you get this expected behavior of a decrease in event number without a change in fluorescence. So, conventional flow cytometry has limitations. Certainly light scatter as a trigger channel is a limitation. It's just really hard to make good light scatter measurements of such small dim. Fluorescence-based triggering, which most commercial instruments are capable of, is probably the solution. Coincidence, as I just described, is a major thing that need to be on the lookout. And then finally, especially in terms of triggering, but also in terms of data reporting, calibration of the fluorescence measurements is really important. And when you're measuring lymphocytes, expressing results as percent positive, it's not the best quantitative description of your data, but it's probably okay because you'll at least know what the denominator is, lymphocyte concentrations. But for vesicles, where no one's really sure about what concentrations are, percent positives don't really mean anything unless it's in terms of some detection limit. So I'm gonna spend a couple minutes here talking about approaches to calibrate your instrument and your measurements. So you can buy beads that have calibrated intensities, and probably most of you run these routinely, or your core facility does. But there are very well-established approaches to calibrate and report fluorescence response. The multi-intensity, multicolor beads are a great tool. That brightest bead is often used for instrument alignment, instruments that still need to be aligned. But actually, most of the information about fluorescence response comes from the dim populations of beads, which can tell you about how many photons you're detecting. And this approach to calibration of flow cytometers has been worked out over about the past 15 or so years. And key measurement is Q, or the detection efficiency. So by measuring beads of differing low dim fluorescence, one can, if one measures the broadening of the fluorescence distribution as a function of brightness, find that the populations get broader as the signals get dimmer. And the main reason for that is photon counting statistics. So when you count small numbers of anything, statistics are Poisson in nature. One can look at the variance of the population distributions and use that to calculate how many photons to detect. And Q is a figure of merit for a flow cytometer or an instrument that has units of either photoelectrons per channel, or if you have a absolute calibrator, a bead whose intensity is reported in units of mean equivalent soluble fluorochemicals, MESF, then you can talk about how many photons you've detected, MESF. There's a little bit of math that's involved here. Many of these slides were borrowed from Bob Hoffman, who did a very nice tutorial at a Cyto meeting a couple of years ago. These are available online for you to view. I'll tell you about that. But basically, by plotting the standard deviation of those bead populations versus their brightness, one can calculate Q, detection efficiency that I just described, and B, the background. And using these two numbers, Q and B, you can understand a lot about how your instruments work. So, okay. So basically, these beads are probably run frequently anyway in your lab. The analysis is slightly complicated. I mean, it's sort of intermediate flow cytometry. It's not beginner's flow cytometry, but it's important to do because it gives you these, these key calibration numbers, and also teach you something about your instrument. It's always good to know more about your instrument. The bottom line in terms of Q is that it really can predict how well you're going to do at detecting small dim particles. So on the right here is an example of an instrument that has a low Q, and you can see while the bright bead looks fine, it's bright, it's narrow in its distribution, the resolution of the dimmer particles is compromised. And so this has direct implications into we're trying to measure small particle type. There's another number that can be calculated from B, and that's resolution limit. This can be thought of as the sensitivity of your instrument. It's a measure of the minimum MESF, minimum intensity detectable with 95% confidence. And so here is for the FACS caliber in our lab, a picture of a graph of what a histogram of what the resolution limit would look like in this instrument. About 2,000 molecules of fluorescence, which is pretty typical in the green channel for instrument. So this is a measurement that everyone can make on their own flow cytometer, and I sort of recommend it if you're going to try to measure extracellular vesicles that may have only a few thousand or a few hundred molecules on the surface. As I mentioned, fluorescence triggering is, I think, gaining favor as the approach to detect these vesicles. Again, Alain Brisson's group did a systematic comparison of EM, fluorescence-based triggering, and light scatter-based triggering, basically detecting 100 to 1,000 times more vesicles by fluorescence triggering than by light scatter triggering. So where we're going here is it's really going to become a sensitivity problem. If the highest abundance molecules are only going to be there at about 5,000 vesicles, it's about as bright as any vesicle might get. It's 5,000 antibodies bright. And most of the interesting biological markers are likely to be much infrequent, rare. We're going to need higher sensitivity instruments. And so the lymphocyte analyzers probably aren't going to cut them. So most of the more sensitive instruments are still developmental systems. They're not actually available yet. Zhao Meiyan's group, Xiamen, China, has a very high sensitivity flow cytometry that uses slow flow and very tightly focused laser beams to measure individual mitochondria. And they've shown individual lipid vesicles carrying various cargo. They can show detection sensitivities approaching a single molecule, which is going to be required. Some rare surface markers that might be of interest. In my lab, we're taking also a similar approach to build high sensitivity flow cytometers. Looks about like a conventional flow cytometer, but we've optimized the laser, the fluidics, the electronics to build it for sensitivity. And so this system, which we're just getting ready to publish on now, has a resolution limit of about 200 fluorescent molecules. So it's about 10 times more sensitive than FACS caliber. The point here not is that this is the end all and be all of instruments, but that we can characterize our instrument and know what our resolution is. That's anyone in this room should be able to do if interested. So we've taken a high sensitivity flow cytometer and we're making vesicle movements. And so the approach we're taking is fluorescence-based labeling. We've got a membrane probe, dye 8 ineps. It's a voltage dye that happens to have the nice properties of being almost non-fluorescent in PBS, but when it gets into a membrane, it greatly enhances its fluorescence. So we use that to detect vesicles, and then we have a green fluorescent antibody label to measure surface. So this is a two-color instrument, but it's a sensitive instrument. The approach we're taking is minimalistic in terms of sample prep. We spin a sample a couple of times to get rid of the large stuff, so we just have the particulate plasma or culture media. We stain with our membrane probe, we stain with our fluorescent antibody, and we analyze them one at a time. With the appropriate calibration protocols, it's possible to express membrane dye fluorescence as a size of a particle. So we can measure the size of particles. And then we can measure the abundance of a surface marker in absolute terms of MES. So here, x-axis here is brightness expressed in MESF. And so this is where I'll leave it. This is, I think, sort of the future of where this area needs to go. We need to have sensitive instruments, we need to have appropriate sample staining, and then we need to have the right kind of calibration to express our results in absolute terms. So just to wrap up, there's a bunch of resources available, most of them for free. Current protocols in cytometry is a great place to learn about how to do some of these calibration protocols. Bob Hoffman did a scientific tutorial at a Cyto meeting. There was just last week a webinar on Cyto University on characterization of instruments. Cyto University is the online education portal for ISAC. ISEV, the International Society of Extracellular Vesicles, is really helping move the field forward and standardize. They've published position papers. I recommend everyone to look at this recently published one. It's just about how you should report on vesicle preparation characterization. Then recently, the three societies I've mentioned during this talk decided to form a working group to try to standardize and provide some uniformity. With that, I'll plug Cyto, which is happening in a month, and take any questions.
And we have limited time, but time for probably one or two questions. And if anyone would like to ask them, please approach the microphone.
So the resolution limit depends on your back and your Q. So the question was, what kind of electronic noise from the PMT is acceptable for measuring these particles? So any noise is bad, any noise is background. There are approaches to determine the optimal PMT voltage for an instrument. Basically involves measuring the same sample at a range of PMTs voltages and looking for the point of diminishing return where you no longer get signal noise. In general, electronic noise probably isn't hitting there in our experience. Certainly there's no value in turning the detector up higher than it needs to be. But in general, if one follows those protocols for determining optimal voltages, it'll be okay in terms of optimizing the response of the instrument. So as I understand it, the question is about what about isotype controls, and where do you set your threshold for positivity of the signal? And there's a lot of debate about use of isotype controls in cell-based analysis. We don't use isotype controls. We don't find them very useful for these vesicle analysis. The background staining of non-specific staining of a vesicle is much, much smaller than that of a cell. And so in general, we find the biggest source of background is not from non-specifically bound antibody, but from free antibody that just happens to be in the background. Because you can't really wash vesicles easily because they're so small, you can't just, it involves dilution. And so really what you have to pay attention to is diluting your antibody enough that you don't have significant signal from free. I'd be happy to talk to you about that. Antibody titrations are always a good idea.
So actually, unfortunately, that's all the time we have today. But you guys are welcome to come back to the booth and talk to us if you're here. And if you're joining us from online, also thank you for attending. We will answer any of the questions that you've typed in via email after the webinar concludes. So thank you very much for attending today.
This is Laura Fulford from Bite Size Bio. Thank you for taking the time to attend and listen into this live webinar today. If you've enjoyed the webinar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours, but you'll get an email when it is gone live. There you can also see the other webinars we have lined up for you in the Bite Size Bio Webinar Festival, and also watch any of the previous webinars that we've held during this festival. So until next time, good luck in your research, and goodbye from all of us at Novus Biologicals and Bite Size Bio.